Hey, Liv. Here comes everybody. It's great to see you all today. What's new? Not too much. All right. Remember, I need all cameras on, at least at the beginning of class. Yeah, that is AIM Academy policy. Your poor cat keeps getting hurt. How's your cat getting hurt? How does it? Cats are known to like land on their feet all the time. It's rare to see a cat get hurt. Allergies. Oh, allergies are tough, no matter what animal you are. And he broke his tail. Like, well, actually broke one of the bones. Wow. That's terrible. Oh, your sister shut a door. Yeah, that would do it. Oh, that sounds painful. That sounds painful. All right. Anybody have any questions for me before we get started? All right. If not, we will jump right in. So if you were able to have a helper, an assistant today, that's great. If not, I will pair you up with someone else who does. Tell you how we're going to do that when we get there. All right. So just real quickly, before we get started with our activity, I want to review the kind of receptors, of touch receptors that we have in, oh, look, there's the cat. Oh, poor baby. Animals are amazing. They're absolutely amazing. Yeah, I'll tell you just a little side story about a cool animal story. One of my neighbors has a dog that he takes to hospitals and nursing homes to care for. You know, it's like he's a therapy dog. So it soothes people, it comforts them. This is a collie, so it's a very big dog. He's even big for a collie. Yesterday they were in the hospital and there was a woman who was crying in the hospital, and this dog, with no prompting from him, walked right up to the woman, touched his nose to her nose, laid his face beside her cheek, and stayed there for about 5 minutes. That's a long time. I mean, do we deserve animals? I think not. I think they're pretty amazing. Anyway, all right, back to the skin. So can you guys all see that? Where the skin pictures? Okay, good. So we've got all these different kinds of skin receptors. If you're keeping up with the due dates, you learned about these yesterday in your lesson. So we've got free nerve endings. That's right here. Now they determine, they get really light touch as well as pressure and some pain and temperature. So they're kind of the do it all. The free nerve endings do a little bit of everything. Then you've got Ruffini. So these are right here. Ruffini sense heat and pressure. Pacinian, these are my favorite. They look like an onion and they determine pressure. So when these little layers here, these layers of the onion, when just the top ones are compressed, they send a message to the brain saying there's a little bit of pressure. When these bottom ones are being compressed, they send a message saying there's a lot of pressure. And then we have Meissner's, and that's what we're going to work with today. Meissner's are for light touch, medium touch, and then superficial pressure. So just a little bit of pressure, not the deep stuff that the Pacinian corpuscle would get. All right, so we're going to test for Meissner's corpuscles. We learned that they're one of the kinds of receptors that we have in the skin, but we also know that they're not evenly distributed throughout the body. So what we're going to do today, this activity is going to show you where there are more Meissner's corpuscles and where there are fewer ones. Now, even taking a look at this drawing, notice how much closer to the skin surface the Meissner's are than the Pacinian. There's the Pacinian, or the Meissner's right there. There's a Pacinian way down here. The Meissner's are much closer to the part that you touch, the surface. So Pacinian tests for deep touch. You have to use pretty light touch to avoid activating this Pacinian in this lab. So you don't want to be pressing really hard with your pencils. All right. Now, think about your entire arm. Let's just think about the arm all the way from the shoulder down to the ends of your fingertips. What do you guys think are the most sensitive part of an arm? You think the skin on your shoulder is the most sensitive? The skin on your palm? And you can kind of tell guys just by touching. Touch your upper arm on the outside. And then touch your forearm right here. Now turn it over and touch your forearm on the inside and feel the difference. Now touch the palm. And now your fingertips. You can tell just by that little practice experiment that the areas of your skin that are the most sensitive are going to be the ones on the inside, the palm, and the fingertips, way more than the outside and the upper arm. All right, so make sure that you have the materials gathered up that I told you to get. So this test that we're going to do is to determine the area of the arm where we are best at determining when there's 2 points of contact as opposed to 1. So we're going to use 2 pencils taped together that look like this. So take your pencils now and tape them together. And they have to be so that the points are even. It won't work if one of the points is way down here and one of them's up there. They have to be as close to even as you can get them. All right, so go ahead and tape your pencils together. Okay, while you guys are taping your pencils together, if you don't have a helper, put your hand up. Okay, so Vida and Anastasia. Anybody else? Lane, Olivia, Rain, Abe. Abe, are you going by Abe this year or do you want Abraham?
Other one is fine.
Okay, all right. I know last year you had said Abe, so that's what I'm calling you. All right, anybody else besides Vida, Anastasia, Lane, Olivia, Rain, and Abe? Okay, so those of you who do not have your cameras on, are you able to turn your cameras on? We'll need to have them on in order for you to be able to help someone else. So I'm going to pair you up with somebody else. All right, so Vida, you will work with Savannah. Anastasia, you will work with Talani. Lane, you will work with May. Olivia, you're going to work with Arwa. Rain, you're going to work with Emmeline. And Abe, you will work with Ben. Okay? So Ben, are you able to turn your camera on so that Abe can work with you? All right, terrific. Oh, you don't have a partner. Okay. All right. So Abe, then we will put you with Talani.
I already have a partner.
You don't have a partner?
I do.
Okay, yeah. So I'm gonna have you help out Anastasia. She'll watch you. Okay, so I'll explain what I mean in just a second with the partners, how this is gonna work. So we need somebody for Abe and Ben. I guess.
No,
right.
I do have a partner.
Oh, you do, Ben?
Yeah, I do.
Oh, okay. Okay, I thought you were saying no. All right, all right. So by partnering, what I'm doing right now is they'll watch you do it and they'll use your data for their chart. Okay, so it's as if they're doing it. Okay, it's better if you can do these, but if you can't, I do understand it's not always easy to get somebody to be able to come and help. Okay, so let's get started. I'll explain what we're doing as we go. Make sure that your pencils are taped together. And these are the areas of the body that we're going to test. So I sent you guys this sheet, this 2-point discrimination lab. And right here is what we're looking at. It's the data recording sheet for 2-point accuracy and 1-point accuracy. If you didn't print the paper out or you have it on your computer, open on your computer, you can just write these down right now. Write down forehead, cheek, forearm, palm, tip of thumb, tip of index finger, and back of lower leg. Okay, now what you're going to do is test each area of the body. We're going to touch an area alternating 1 point sometimes and 2 points other times, and you're going to keep records as you go. So you'll touch and your helper will tell you whether they feel 1 point or 2 points. Now, for those who are not able to do it, who don't have a partner, you're going to work with the person that I told you their name. or somebody who raises their hand, it's fine. So whoever I paired you up with. So for instance, Savannah is going to do it, and Anastasia or Avita is going to observe Savannah. So Savannah will simply hold up the fingers, telling us whether it was 1 point or 2, and then thumbs up if her partner got it right, and thumbs down if her partner didn't get it right. Okay, so those of you who are, so Savannah, Talani, May, Arwa, Emmeline, and Ben, you guys are gonna, when you're doing 2 points, you're gonna hold your fingers up somewhere that your partner can't see, or 1, whatever where you're touching, and whether they got it right or whether they got it wrong. Okay, does that make sense? Hopefully it will as we continue going on. And you're gonna write this down on your record sheet, on that sheet that I gave you. Now, the more Meissner's corpuscles there are, the more sensitive the area is gonna be to touch. So at the top of your sheet, you have this right here, the lab question and the prediction. So let's talk real quickly. What's our question here? What's the question that we're answering with this lab? Anybody? Oh, what are we trying to find out? We're trying to find out which are the most sensitive. So our lab question might be, if we tested these areas, which will be the most sensitive to touch? And then our prediction, what we're going to predict. So we might predict that the fingertips have the most sensitivity to touch. We might predict that the back of the lower leg has the most sensitivity to touch. That would be our prediction. Then the next thing on our lab sheet is a hypothesis. Now remember, whenever we possibly can, we want to write a hypothesis in what we call if-then format. If something is done, then something will happen. So there's a couple of different ways we could write these hypotheses. We could say, if 7 different parts of the body are tested for sensitivity, then the palm of the hand will be the most sensitive. Back of the leg will be the most sensitive, whatever we predicted. We could take it a little bit farther and say, if 7 different parts of the body are tested for superficial pressure and superficial touch and superficial pressure receptors, then the what will be the most sensitive. Now, the idea here, when you write it in if-then format, if is the independent variable, the thing that you're changing. You're changing where you're touching. Then is going to be the dependent variable. Dependent is what you're measuring. Independent variable is the one you change. Dependent one is what you measure. Controls stay the same, whatever. Okay? All right, so we're going to all work together to do this. And then we're going to use this chart. Now, if your partner correctly identifies 2 points on the forehead, you're going to put a check mark right there, like that. See, 2 points, forehead, they got it right. You put a check mark. If when you did 2 points on the forehead, they told you there was 1, you're going to put an X. All right? If your partner correctly identifies 1 point on the forehead, you're going to put a check mark. If they correctly or incorrectly identify it, so you touched 2, or you touched 1, but they said 2, then you're going to put an X. Basically, if they get it right, put a check mark. If they get it wrong, put an X. All right. Now we're going to try at least twice for each body part with 1 point, and at least twice for each body part with 2 points. All right. Now, have your helper sit down in a chair and blindfold them. Or if you can absolutely trust your partner not to peek, you can have them just close their eyes. But it's sometimes hard not to peek. It's important in this experiment that you use light touch. First of all, you don't want to hurt your partner with pencil points. Second, you don't want to activate those Pacinian corpuscles. We only want to be activating the light touch Meissner's corpuscles. So basically, you're just touching just until it comes into contact with the skin. No pressure at all. So once they're blindfolded, we will start with the forehead. We'll do 2 points and 1 point. Now you don't do them in order. You can do 1 point first or 2 points first. The idea here is you don't want it to be predictable. So you're going to gently touch the pencil points when you're doing 2 at the same time. And you're gonna ask your helper, did you feel 1 or 2? And then you're gonna put 1 if they felt 1 and thumbs up if they got it right. Thumbs down if they didn't. All right. Now remember that you don't want to push too hard. The idea here is to find out whether your helper can tell the difference between 1 point or 2. So you should test 1 point and 2 point both on the forehead, but mix it up a little bit so that they don't see a pattern. You don't want every time you start on a new body part to use 2 points. They'll get the idea. So sometimes use 1 point first, sometimes use 2 points first, and you want to put a check mark on your chart with what they've done. All right, so let's do the forehead together. Again, only activate Meissner's. Do not press hard enough to activate the Pacinian corpuscles. All right, so do 1 point and 2 point on the forehead with your partner at least twice for each. So you want to touch at least twice with 1 point, at least twice with 2 points, and tell whether you got it right or not. We're only doing forehead right now. Okay, begin. Okay, Savannah is telling you what she's doing there. So those of you, Savannah, Talani, May, Arwa, Evangeline, and Ben, you guys want to be, Emmeline, I'm sorry, and Ben, you want to be holding up your fingers, how many points you're doing and whether they got it right or not. So those of you who are watching can write it down. All right, keep going until you've done them all, till you have tested all the areas, and then either just stop and hold still and look at me or wave your hand or something so that I know when you're done. Make sure to thank your helper. Think how brave they're being to let you come at their face with sharp pencils while they're blindfolded. A lot of bravery there. All right, when you're finished with the forehead, now we will move on to the cheek. So somewhere in the cheek area, and you can go back and forth from one side to the other. Just make sure that you alternate. You know, you want to do at least twice with 1 point, at least twice with 2 points on the cheek, and write down your check mark if they got it right and your X if they got it wrong. Okay, we ready to move on? Okay, let's move on now. The next one is the forearm. Now I'll leave it up to you. If you want to do the outside of the forearm or the inside of the forearm, just make sure you write it down. Put in parentheses after forearm, inner or outer, because there's a difference between the two. So, or do both if you want to. Again, at least twice with 1 point and at least twice with 2 points. All right, so we lost Emily, so Rain, you need to be working, I guess, watching somebody else. Okay, now let's do the palm of the hand. At least twice with 1 point, twice with 2 points. Now, those of you who don't have partners, you're getting the idea of what we're doing. You could do this later today if you want to actually do the experiment yourself. You don't have to. You don't have to submit these sheets. This is not, going to be homework, but it's fun to do. All right, now let's move to we did the palm of the hand. Let's move to the tip of the thumb. Remember to use very light touch, only activating those superficial Meissner's corpuscles. Okay, when you finish tip of the thumb, go right to tip of the index finger. We're going to skip back of the leg for now. You can do that on your own. If you finish up, you can do it if you want to. When you finish tip of the thumb, move into tip of the index finger. Don't push too hard. Only the Meissner's corpuscles. So the word corpuscle basically just means little tiny part of the body. Sometimes blood cells are called corpuscles, but these little bitty touch receptors are often called corpuscles too. Okay, let me know when you're done. We have one more thing before you release your helpers. One more thing we're going to do. All right, I'm going to show you what we're going to do. The last thing we're going to do with our helpers is this experiment here. I thought she was broken. No, that's pretty typical, pretty typical. All right, so this is extra. This isn't going to be a part of our lab report, but I want you to do it anyway. So putting both points, 2 points right here on the upper inner arm, kind of up near the armpit. You're going to very slowly, like in the little animation I have here, very slowly, you're going to run those 2 points down the inner elbow, down into the palm of the hand, all the way to the tip of the middle finger. Your helper is going to tell you as soon as they feel 2 separate points instead of just one. When you touch the up in the arm, they're only going to feel 1, even though you're touching with 2. As you go down, there's going to be a point where they're going to say, oh, now I feel 2. That's where you want to know. Let them tell you where it is when they just feel 2 points instead of just 1. And then when you're finished with that, your helper's done. We'll talk through this after you release your assistants. Now, after class is over, might be fun for you to repeat this experiment on yourself. Have your helper do all the touching on you. Right about there, Savannah? Yeah. All right. Anybody still working? Sometimes I have fun with the animations on PowerPoint, so this one was a fun one. All the way down. All right, so let's discuss this now. Thank you to the helpers. Appreciate it. If anybody isn't done yet and needs a little more time, let me know. If not, I do want to discuss this a little bit. So let's talk about our data, because basically what we've been doing here is the pieces of a lab report. A lab report has your question in it. What is it, or purpose? You can think of as the purpose or an introduction. What were we trying to do? We were trying to figure out where most of the highest concentration of these Meissner's corpuscles are in the body. Our prediction was wherever you think. If you think the highest amount is going to be on the upper arm, you know, or on the forehead, then put forehead. That's your prediction. Then you put it into if-then format with your hypothesis. What are you going to do and what are you going to look for? So the hypothesis could be something like, when these 7 body parts are tested, this one will have, you know, or if these 7 body parts are tested, then this one will have the most sensitivity. That would tell us. Your supplies. What are the things that you needed in order to do this experiment? When you do a lab report, it needs to have every single supply that you needed. You need to write down helper and blindfold and even chair for the helper to sit in, right? You need to make sure you have pencils and tape, paper, your lab sheet to write things down, everything that you use. And then your procedure, you would write down what you did step by step. We're not doing this. This is not a lab report. We're just talking through it now. But you would write every single thing that you needed. So that supplies and procedure part needs to be so detailed that someone who knows nothing about the lab could step in and reproduce it exactly the way you did. So if you skip tape in your supplies list, they're not going to be able to do it because they're not going to be able to tape the pencils together, right? You have to have everything in both your supplies and your procedure. So now what's our data? Let's talk about our data. We'll go back here to our chart right here. So where was the area that was the least sensitive? In other words, you got the most X's. What part of the body would you guys say are the of what you tested are the least sensitive? Meaning the person that you were testing got them wrong. Where did they get it wrong mostly? Forearm, forehead, forearm and forehead. Most X's on the forearm. All right, we're seeing a pattern here now. Clearly, the forearm is not as sensitive. Now, those of you who had forearm, did you test the inside or the outside? Inside? Outside, a little of both. Got both here. All right, inside, and it was pretty sensitive. Inside. Palm of the hand. You found the palm of the hand to be the least sensitive, May? Yeah, somebody said the cheek. Yeah, your cheek's not all that sensitive, really. Touch your cheek real lightly. And now move to your lips. Lots of Meissner's corpuscles in the lips. There's lots of all the different kinds of touch receptors in the lips because that's an entry point to the body. So your body is going to be careful what it lets in. It wants to be very aware of sensitivity around your mouth. All right. What was the most sensitive of the places you tested? Meaning, where did they get it right? Tips of the fingers? Fingertips, index finger. Anybody see much of a difference between index finger and thumb? Not much. All right, so when you did the running down the arm, where did they start feeling the 2 points? So right here it started, you could only feel one. Where did they start feeling two? If you said it already, it went by. All right, near the elbow, right down the elbow, about an inch down, right above the elbow. Immediately, so we had one right away, she could feel two. Some people have more. Yeah, it is. It is possible to be 100% correct. Usually not. It's pretty rare. 2 inches down from the armpit. So that would be about there. All right, so we got a little bit of variety there. In general, most people start feeling 2 points right around the elbow. Now, part of the thing here is that your helpers knew, because they could hear me say it, they knew that you were touching them with 2 points. So when you put that idea in their head, they might have felt the 2 points, but not really felt it. You know what I mean? Suggestion is pretty powerful. So they knew there were two, so they were more likely to feel two. Yeah. If you had said, you know, tell me what you feel without telling, that it might have been different. All right. So back to our lab report that we were looking at, the lab report sheet. So we had our data. We did our analysis and conclusion. So what could you conclude based on the experiment that we just ran? Where do you, you know, what areas of the body have the most Meissner's corpuscles? That would be your conclusion. Based on, you'd say something like, based on the results of this experiment, the area of the body with the most Meissner's corpuscles is, and for most of you, it was the fingertips. The area with the least amount of them, for most of you, it was the forearm that you tested. Pretty interesting. So you can do this later with other body parts too. I think that's an interesting thing to do. All right, anybody have any questions about this? Just know all of these touch receptors, they're differently concentrated in different areas of the body. All right, that's it for, go ahead, Anastasia.
I know some people have like are just more sensitive to like their environments and stuff, more bothered by like scratchy fabrics and stuff. Is
Yep.
it because they have more receptors in their skin?
So it's a very complex answer to that. It could be anywhere along the chain. It could be that they are more that they simply have more receptors. It could be that their receptors are closer to the surface of the skin. It could be an issue with the skin itself, not the receptors in it, but the skin itself is more drier maybe, or has you know, some different kind of sensitivity. It could be a connection issue between, remember, from the corpuscles all the way to the brain, they have to go through a chain of nerves. Maybe there's some extra sensitivity in the nerves that go all the way up. It could be in the area of the brain that's interpreting that signal. You know, sometimes people feel pain. You know, I had this happen to me the other day that I was looking at my arm and it was itching like crazy. And I'm looking at it going, there's absolutely nothing there. What's making me itch? I have no idea. It could have been that there was some little trigger in my brain that was making me think I had something on my arm when in fact I didn't at all. So anywhere along the way, there could be something. Sometimes even, so hormones can play a role. You know, the activity of hormones can play a role. Yeah, it's, this is why when I say that it's really hard because we have to study the parts of the body separately, we are so all connected. We just have to remember that too. Yeah. Does it have to be skin related? No, it absolutely does not have to be. I get overstimulated a lot by sounds and textures. And yeah, it could be. So one of the issues with autism is that when a person is autistic, a lot of times their brain doesn't have the ability to filter things out. So like, let me give you an example right here. If you're wearing a pair of shoes right now, until I said that, you were not aware of your shoes, but you are now. Think about your shirt that you're wearing. Now you're aware of it. A minute ago, you weren't. When someone has autism, often they are always aware of their shoes, always aware of their shirts, always aware of the sounds that come from the outside. So it doesn't have to do with skin. That's a brain issue and not a skin issue at all. Yeah, good question, Lane. All right, real quickly, I want to go over these anatomical directional terms because they are so important. We're going to hit these from time to time as we go through this. So either type in or yell out the answer. Here's the word bank down here. These are the words that we're going to use. In the anatomical position, the face and palms are on the what body surface? Face on palms. What word from that word bank would I use there? Yep, anterior. It could use ventral. You could use ventral, but in this case, we're going to say anterior. Ventral often has more to do with the belly, where anterior would be front. But both of those terms could be used for that one. So we'll use anterior. And the buttocks and shoulders are on, what's the opposite of anterior? Posterior. Very good. All right, the top of the head is the most, what part of the body? Superior. Superior. Think of something being superior as being higher. So something that would be superior. Now, in a dog, the most superior part of their body is going to be their spine or their head, right? But in a person who's standing upright, it's the top of our head. The ears are what to the shoulders? So here's the ears, here's the shoulders. They're going to be medial to the shoulders, more toward the middle. The ears are, well, I've got superior. All right, sorry, but they are medial. The ears are going to be in toward the middle from the shoulders. So the ears are superior to the shoulders, and then they're going to be lateral to the nose. All right, lateral means to the outside. Got your nose here, lateral to the outside. The heart is what to the spine? More toward the front of the body. Anybody? It's going to be anterior, anterior to the spine. And what to the lungs? So the lungs are on the outside of the heart. So it's going to be more toward the middle. So that would be medial to the lungs, medial to the lungs. Ever so slightly posterior, Liv. Yeah, ever so slightly, the lungs go a little bit back behind the heart, but for the most part, they're just going to be, the heart is going to be in the middle of the lungs, so medial. All right, the elbow is what to the fingers? Now we're talking anatomical position. So in anatomical position, where is the elbow in relation to the fingers? So kind of, you can kind of say superior. We probably, yeah, proximal would be the word that we would use for this. Proximal. Proximal means closer to where it attaches to the body. where distal means farther from where it attaches to the body. So we wouldn't really use superior for the arms and legs so much. We're gonna use proximal and distal. So the elbow is proximal to the fingers, but distal to the shoulders. Now in humans, the dorsal surface can also be called the posterior surface. So dorsal and posterior both mean the same thing in people. In four-legged animals, dorsal is near the top, so that would be superior. All right, like I said, we're going to go over those and over them and over them because you need to know all those terms. I recommend printing those terms out, and I gave you guys a chart. There's a chart in the course resources on the homepage, and putting it in your notebook and having something real nearby. All right, now we're going to lighten things up a little bit. We're going to do something that's just a little bit silly. So we're going to miniaturize ourselves for injection into the skin of our host, and we're going to go on a little journey. All right, so our journey begins when we are deposited in a soft, gel-like substance. Our immediate surroundings resemble huge, grotesquely twisted vines. When you look closely at the nearest vine, you realize you're actually seeing some tissue fibers. What are the 4 different kinds of tissues in the body? Only 4. What are they? I know you know them. Good, Anastasia. Connective, epithelial, muscle, and nervous. Remember, if it contracts, it's muscle. If it carries a message, it's nerve. If it covers or lines the body, it's epithelial. If it's not any of the other 3, it is connective tissue. So what kind of tissue are we looking at right here? With these twisted vines. Yeah, this is going to be a connective tissue. Very good. All right. Now, you see fibroblasts right here. What do they do? What do fibroblasts do? Anybody? Remember when you hear the term blast, think the cells are having a blast making things. They're just having so much fun making them. Yeah, Liv, you got it. Make the fibers. So fibroblasts are having a blast making fiber. So fibroblasts secrete collagen that's needed in order to build those fibers. Now, the opposite of that would be blasts. Would be not the opposite. Another kind of blast would be like an osteoblast. Osteo means bone. So osteoblasts have a blast making bone. But in this case, these are fibroblasts. They are making these fibers. They're secreting the collagen fibers. So even though they're tangled together, most of the fibers are fairly straight and they look like really strong cables. So these are going to be collagen fibers. Now here and there, there's thinner fibers that are going to coil and twist. What are those thinner fibers? If they're not collagen, the thicker ones are collagen. Let me get my pointer here. The big thick ones right here. These are collagen. Yep, Anastasia, good. These thin ones are going to be elastin. So those elastin fibers are going to help to give the skin its springiness. When you pull up your skin on your hand, you can pull your skin up away. That's because there's elastin in it. It's that springiness that is because of the elastin fibers. So there we have collagen and elastin. All right, now we look around and we can see blood vessels and nerves around us. So what layer of the skin are we in if we can see blood vessels and nerves around us? Remember, we got those different layers of the skin. What one are we in right now? Yep, we are in the dermis. So carefully using those collagen fibers as steps, we begin to climb upward. Now, after we climb for a little while, we find that we still haven't reached the upper regions of the skin, but we're going to stop right here and rest a little bit. Now, as we sit there, a very strange looking cell approaches. It gets close to us. It's moving slowly with alternately flowing and receding parts. Anybody have any idea what kind of cell that is that's kind of moving through the skin? We haven't learned about it yet, so there's no reason you should know. But this is a white blood cell. White blood cells move through the skin to get rid of intruders. And it's seeing us right now as an intruder. This kind of white blood cell is actually called a macrophage. And this is what it would do to us if it caught us. Take us right in. This actually is the way that macrophages work. So we need to get out of there. We need to run. So we're going to scramble to our feet and we're going to resume our upward climb. Now on our right is a large fiber. It looks a lot like the trunk of a tree that's anchored in place by muscle fibers. What is this called? This whole thing. What is this called? Yeah, it's a hair. The hair sticks out of what's called the hair follicle. So the hair follicle. Yeah, you got it. Now, by scrambling up that hair follicle, we're able to get away from that white blood cell. Now, once we're safely out of harm's way, we can look around for our surroundings. So what are those little muscles that hold the hair fibers in place called? You better remember what the name of those tiny muscles is. They are called erector pili muscles. They erect the hair. They make the hair stand up straight. All right, so directly overhead are tall cube-like cells that form a sheet, a continuous sheet. And that layer is called the epidermis, the layer that they form. Now, in our rush to escape, we have gotten to the epidermis layer of the skin. As we watch the activity of the cells in the bottom layer, we notice that many of them are pinching in too, and their daughter cells are being pinched upward. So this right here, this bottom layer right here is cellular. That is where cells are being made. New cells are being produced. The daughter cells are being pushed upward until we get to here, to these layers that are the stratum corneum and the stratum lucidum are filling up with keratin. They're tough. They're dead. There's no activity in them. The activity is down here in the cellular layers of the skin. Now, looking through the transparent cell membrane of one of these basal cells, we see a pretty dark mass hanging over the nucleus, right there. Zooming in, it looks like this. We wonder if the cell could have a tumor. But then looking through the membrane of the neighboring cells, we find that they also have these dark umbrella-like masses hanging over their nuclei. Anybody have any idea what those dark masses are? Yeah, those little granules. What are those? That's actually melanin. Melanin. So as we're thinking about it, this dark cell with long tentacles begins to pick its way carefully between the other cells. One of those transparent cells engulfs the tip of one of the black cell's tentacles right here. So it just engulfed the tip of that. Within seconds, this black substance appears above the nucleus. So there's the nucleus right here. So step A, it's engulfing it. It's pinching it off. Within seconds, it begins to form over the nucleus of the cell. So now we remember one of the functions of the skin is to protect the deeper layers from sun damage. This black substance must be that protective pigment called melanin. So if this is melanin, what is this cell called? What is this type of cell called? Yep, it is a melanocyte. Very good, very good. All right, once again, we begin our upward journey. We notice that these cells are becoming shorter and harder, and they're full of that waxy looking substance. I just said it a minute ago. What are they filled with? What is the protein that fills up those outer layer of cells? Keratin, right, right. Keratin is why these cells are getting tougher and harder. All right, now climbing higher still, these cells are flattened. They look like really like shingles. So the only material that's apparent in them is that keratin. No nuclei, doesn't look like there's any activity in those cells at all. These are squamous cells. So we know that we're very close to the surface of the skin. Suddenly we feel a strong rumbling in our immediate area. The pressure is tremendous. Looking up through those transparent cell layers, we see our host's fingertips vigorously scratching the area directly overhead. We wonder if we are causing her skin to tingle. So we end our journey and we run for it. Is there DNA? No, Anastasia, great question. There's nothing in there but keratin. The ones that are on the outside are completely dead. There's no DNA at all. Good question. All right. Can dead cells be used for identification? No, dead skin cells, unless there's something. So like when they're doing DNA, looking for DNA at maybe like a crime scene, they're looking for things like hair follicles. They would be looking for pieces of blood. They can't use dead skin cells. Yeah, thank you. I had some fun making that. All right, I want to end today by talking about some of the differences between male and female skin, because there are significant differences. So let's look at individually. Male skin is typically about 1/4 thicker than female skin. So they've got more cells, and those cells would be a little bit thicker. Now that's usually due to the effects of testosterone. The thickness makes male skin more resilient. It's not fair, but it's less prone to getting wrinkles. Male skin is less likely to wrinkle than female skin, less likely to get those fine lines at earlier ages. Female skin is usually thinner and more delicate, and it ages a little bit more quickly. All right, texture is often different. Again, this is primarily due to collagen. Male skin is tougher and firmer than female skin is because of collagen, because male skin contains more collagen, giving it a tighter appearance. So again, usually they show signs of aging later, but once aging starts in a man, it usually happens more quickly than it does. Where a female is going to start aging sooner, but it takes a lot longer. Once a male starts aging, he's going to age more quickly. All right, so oil production. Males produce a lot more oil, again, because they have higher levels of testosterone. So that's part of what makes them age more later, because their skin is naturally oily. On the downside, males are more prone to acne during adolescence and early adulthood than females are. So that oiliness in male skin tends to continue through life, but it also is pretty protective against drying out, against dehydration. Where female skin has a lower oil production, making them less prone to acne. But women often have dry skin, especially after menopause. When estrogen levels drop, women's skin tends to get even drier. Pores. Men have more pores and they have larger pores. So that makes their skin appear rougher, and it can make them more prone to acne and things like blackheads. Women have smaller pores. Sometimes as women change, and especially during pregnancy, a woman's pores may enlarge. They may become a little bit more obvious. Of course, we know that males tend to have thicker facial and body hair. Again, this is due to the effects of the hormone testosterone. Women produce testosterone too, but not in nearly as high amounts as men do. So this makes it so that men have to often shave their faces, and that can cause some trouble to the skin too. Where women's hair tends to be finer. They often will have the same amount of hair on their face, but it's very fine. It's not that rough hair. The aging process, like I said, male skin ages more slowly than female skin, but once it starts, it goes quickly. So women's skin tends to show the signs of aging earlier, especially after menopause, because that's when estrogen production drops and collagen production drops as well. A lot of this has to do with how much collagen you have in your skin. pH, women generally have a higher skin pH than men do. And pain and sensitivity, women tend to be less sensitive to cold, where men are less sensitive to pain and temperature extremes. So that's something definitely different. And then hydration. Because of their extra oil production, men can hold moisture, so they're not as likely to dry out, where women's skin tends to be drier. So women tend to have to use moisturizer more often, especially as they age, and especially, like during and after menopause. Yeah, Vita, the skin does start getting drier after menopause. Sometimes once menopause ends, some of these problems kind of level out, sometimes not, but yeah, a lot of differences there. All right, anybody have any questions? We went a couple minutes over. I hope that's okay. Talani.
When you pluck your hair, I heard that the hair grows into an ingrown hair.
So it can. What ingrown means, let me go back to a picture here. Here, let's look at this one right here. So ingrown basically means that one of these hairs has curved back in and started going back into the skin. That can happen. It's not necessarily from plucking. Once you pluck a hair and it's out, it's gone. When you pluck hair, sometimes you will pluck all the way down and take the whole hair follicle out. And that means that hair will never grow back. So there was a time like in the 50s where women were plucking their eyebrows and they didn't grow back. They've had to live the rest of their lives with no eyebrows at all. Yeah. So you don't want to pluck. You don't want to pluck the hair on your head or your eyebrows. But yeah, they're two different processes. The ingrown, it can be pretty painful though. It gets a little bit infected sometimes when you have an ingrown hair. Fortunately, it's not all that common. Good question. Anybody else? All right. Good participation today. Thanks, everybody. Have a great week. I'll see you next time. Bye.